member of Indian Immunological Society, Society of Toxicology India, Indian Science Congress Association, Association for Pharmaceutical Teachers India, Laboratory Animal Scientists Association, Academy of Science for Animal Welfare. We welcome you, sir. With these words, I would request sir to deliver his deliberation. Please, sir.
Thank you, Ruchi. And I am very much thankful to Professor Amit Rai and management of Columbia Group of Institutions for giving the opportunity for this STTP training programs sponsored by AICTE. And good afternoon to all the participants. And now we will start. Ruchi, is my slide is visible?
No, sir.
Ruchi,
Yes, sir.
is it visible now?
Sir. No, sir, as you have not yet, sir.
Uh, yes. Is it visible?
No, sir. Slide is not visible, but it's showing that you are presenting something.
From any audience side, is it visible?
So there is a response that slide is visible.
Okay. Let's start now. So, in this topic, it is a very common topic for all the pharmacy professionals and the students and the teachers working in the field of pharmacy, that is techniques in experimental pharmacology and animal handling. And we must know about how to handle the experimental animals, because we all people's work in the field of teaching of pharmaceutical education, we all deals with the experimental animals. So in this topic we will discuss techniques in experimental pharmacology and animal handlings from the ancient time periods. We will go to the ancient historical time periods. We can observe that the Aristotle before 384 to 322 BC revealed the anatomical differences among animals by dissecting them. Aristotle before Christ, and later Galen also first the person to demonstrate the living animals, specifically guinea pigs. And William Harvey published his work on heart and movement of blood in animal in 1628. And in France was the leading center for experimental biology and medicine, marked by the work of such scientists like Francois Magendie in experimental physiology, Claude Bernard in experimental medicine, and Louis Pasteur in microbiology and immunology. Gradually, it was developed, means use of animals, biomedical research gradually increased. And in India, the renowned person Sir Ramnath Chopra was the beginner of the pharmacological research of traditional drugs. So from these slides, we can observe that the ancient time periods, from the time of Aristotle, Galen, William Harvey, the peoples were used to animals and for their discovery and research. And why will use in animal in biomedical research? Why animals are used in biomedical research? It is important things. All of we think that कि हम animal क्यों use करेंगे? It is very common questions in the brain of the peoples or the researchers and new students. Students generally ask the teachers, why, sir, why we will use mice or rat in experiment as experimental animals? One of the data indicate that mice shares more than 98% of DNA with us. More than 98% of DNA with us. And another things, teach all these animals, all these experimental animal like rat, mice, guinea pig, having same health problem like human beings, like cancer, diabetes, heart disease, TB, flu, and asthma. As these animals suffering with same disease pattern, same health problem, so we can induce disease on that animals and we can evaluate our drugs. And with a sort, another most important thing that the human is having more than 80, 90, 70 years of lifespan, but it is not possible to do work on that human being, and the life is also valuable. So in that conditions, we can the lifespan of the mice, rat, rabbit, and guinea pig is very small in comparison with human being. So we can select them. We can select them. And another most important important thing is that we can evaluate that disease process. Suppose we have studied some toxicity studies or chronic toxicity studies, and in that condition we have to study for 2 years, 3 years, or another things. अगर हम till end of the lives, if we want to study the effect of drugs for 2 years, 3 years, so entire lifespan we can study by using animal model, but it is not possible for other large animal or human being. So that first thing, very similarity with humans, that more than 98% DNA similarities, more than 98% DNA similarity, same health problems like cancer, diabetes, heart disease, many more, arthritis, and TB, flu, asthma, etc., etc. And another thing, how a disease process is progress, it can easily studied by using that small animals, and how a drug molecule interact, how it interact with our body and living biological system, we can easily study by using animals. Another most important thing that कि कभी कभी यही बात आता है कि हम इतना एनिमल को किल कर रहे हैं, कभी कभी हमारे माइंड में भी आता है कि वी थिंक दैट हाउ नंबर्स ऑफ एनिमल्स आर सेक्रीफाइस्ड इन द बायोमेडिकल रिसर्च. CPCSA make rule to regulate the use of animals. So this question, if you will observe the literatures and some books, that household killing of animals, household cats kill approximately 5 million animals every week, and more than total number of animals used in biomedical research. If our social activists will observe this data, that more than 5 million animals killed every week, and we use less than 0.2% research animals in comparison with this data. So use of animal for research, for development of science is very less in comparison with unnecessary killing by household cat. So this is the status of the animal use across the world. It is very much essential data we should know. And another things, for the development, the experimental animals are very much essential and very much boon हमारे लिए वरदान जैसे हैं, क्योंकि development of any drug molecules, if you want to develop anesthetics, you want to get tetanus vaccine, you want to develop penicillin, you want to develop insulin, all the research are carried out on, because all these drugs are very much essential to us. Insulin is essential to us, anesthetics is essential to us, tetanus vaccine is essential to us, very much essential. And another most important, not only the use of development of the drug molecule, but also development of some instrument like CT scan, MRI. We also use animals for their evaluation, for their developments. So animal use of animal is not only essential for developmental drug, but also it is essential for development of instruments also. Much many more instruments. There are trials carried out on animals, and its efficacy calculated. Then we studied on human being in different phases of trials. So in next phase we will discuss some commonly used experimental animals. I think all of we know which animals are used, uh, and why it is used. Most of the pharmacy professional know that mouse is very, why will use mouse, and all of we know that about mouse is the smallest among all experimental animals. Smallest. Why we will use mouse? Because it is very small, it consume less amount of drug, and it can handle easily, highly exploring and mobile, and easily we can administer drugs, and the expense is also very less to maintain the mouse. The maintenance cost is also very less, so we can use mouse as an experimental animals. I think all of we know and we have seen mouse. Another animal is rat, Ratus novargicus. It is very commonly used experimental animal. Weight is about 100 to 150 grams, and they can withstand long period of experimentation under anesthesia. But in comparison with mouse, a rat can easily withstand the pressure condition, and they can easily handling a mouse, handling a rat is very easy, and drug can be injected intravenously, subcutaneously, intramuscularly, intraperitoneally, and they can also drug. We can also give the drugs orally. Isolation preparations can be taken from them easily. We generally in our departments to perform some isolated tissue preparation. We work on some isolated tissue like stomach, fundus, colon, uterus. We can easily isolate from the rat. So all these regions. Anybody is to, if we are familiar with the handling of animals, we can easily handle rat. We can easily handle mice. So and it is easy to maintain them. And this animal also required less floor space. इन लेस स्पेस हम एक ही छोटे से कमरे में 500-700 एनिमल रख सकते हैं. सो वी कैन यूज these animals. Another thing, rabbit, Oryctolagus cuniculus, and most common strain is New Zealand white rabbit, generally used, and it is used for screening of different diseases like diabetes, diphtheria, tuberculosis, cancer, and heart disease. Another, guinea pig, Cavia porcellus. It is very much docile among all animals, and it is herbivorous. This animal eats green foods, seeds, roots, ready-made chow diet. Another thing that this animal unable to synthesize vitamin C, and vitamin C is very much essential for this animal. Most of the people do not know that this animal need external sources of vitamins. If we are maintaining animal house facilities and we have keeping guinea pig, we should give vitamin C from external sources. Otherwise, it will be infected and it may die. To maintain the guinea pig facilities, we should give vitamin C from external sources, and this is very much suitable model for enteric amoebiasis, hypersensitivity reaction, and immune response, anaphylactic shock, encephalomyelitis, tuberculosis, and ascorbic acid metabolism. This animal is most suitable model used for evaluation of amoebiasis, hypersensitivity reaction, immune response, anaphylactic shock, encephalomyelitis, tuberculosis, and ascorbic acid metabolism. So our main topic is animal handling. Now we will discuss about most important thing that हम जब भी एनिमल हैंडलिंग करते हैं, we should be regularized in our work. Means we should be regular in this in the field of animal handling. If we will stop it, हम क्या है कभी कभी एनिमल हैंडलिंग को बंद कर देते हैं, तब हमको क्या होगा नेक्स्ट टाइम we would our hand, and we will fear to that animal. Gradually anxiety develop. So first things, when you will go for handling of any animals, you should be confident and relaxed. फर्स्ट थिंग यू शुड बी कॉन्फिडेंट. क्या होता है, हम कभी देखें स्टूडेंट्स हो या कुछ फैकल्टीज हैं, जब भी हम एनिमल हैंडलिंग के जाते हैं कि हमको डरते हैं, भाग जाते हैं. बट वी शुड बी कॉन्फिडेंट एंड वी शुड रिलैक्स. एंजाइटी कंडीशन we cannot handle. First, confident and relax. And another thing, regular practice. Regular practice. So in that condition, animal also becomes stress free, and we also become stress free. We can, anxiety will not develop. Most animals have sharp claws and prefer not to. Another mistake we always perform, we always placed the animal on smooth surface and not the hard surface, smooth surface, and that area should not be slippery. That area should not be slippery because the rat and mice having sharp claws and not placed on slippery surface. And we should place on case top or non-slippery linear for bench. We should not placed on slippery surface. And with animals, most species animal easily restrain and handle. With this practice, if we will practice regularly and we place the animal on smooth surface, not slippery surface, we can easily handle. And the gen should not, another thing, most important thing is that uh, during handling we should not give any type of discomfort, any type of pain to that animal. Our handling technique or our handling method should not give any type of pain, any type of discomfort to that animal. And we should concentrate on our handling work. So here this slide is representing handling technique for mice. If we will observe this slide from left to right side, first thing, move the mouse from the cage by grasping, picking its base of the tail, base of the tail. Here, this in this figure, we removed the mouse by, here this is the base of the tail, and by holding its base of the tail, we remove from the cage and placed on the lid of that cage, and then quickly. Another thing, we should be quick in our duty, in our work, quickly and firmly, but not hardly, firmly. माने हम उसको आराम से लेकिन जल्दी से उसका बेक जो नेक पोर्शन है एंड बिहाइंड द ईयर एंड नेक पोर्शन, उसको हम होल्ड करेंगे विद द हेल्प ऑफ थंब एंड इंडेक्स फिंगर. थंब एंड इंडेक्स फिंगर, विद द हेल्प ऑफ थंब एंड इंडेक्स फिंगर, वी क्विकली बट फर्मली, आराम से उसको हम पकड़ेंगे. देन we can easily handle the mouse. It is very much essential. Otherwise, it will cut us, bite us. If we will be, if अगर आप concentratively नहीं पकड़ेंगे, हम quickly नहीं पकड़ेंगे, then that animal will move and bite our fingers. Another thing, if transfer the tail from the preferred hand between palm and little finger. Here the figure number 4 representing. Then we can inject or we perform any other works. So how we will handle? First thing, we remove from the cage by holding its base of the tail, not the tip of the tail, base of the tail. And then we will place on the surface of lid, then firmly and quickly picked up by the back of the neck behind the ear, and then transferred from the preferred hand between the palm and little ring fingers. Then mouse is restrained. This is the best way for restraining mouse. And another thing, another technique for handling of rats. Here, what we will do? Grasp the base of the tail. Always we should grasp the base of the tail, and with one hand, and quickly we should hold the back of the neck by using all fingers. In one method, grasp the base of the tail and grasp the back of the neck by using all fingers. Another technique. And in another techniques, place your index and middle finger alongside In another technique, we can different people's performs holding up animal by different techniques, but your technique should not produce any pain to any animals. Place your index and middle finger alongside the rat's head and your thumb and ring fingers under its forelegs. Here, importance. Index and middle fingers, and all of we know the index and middle finger to hold its head. Index and middle fingers used to hold its head. And next, handling of rabbit. How we'll handle rabbit? For transferring from one case to another, rabbit is having comparatively larger size in comparison with rat and mice, and maybe 2 to 5 kilogram weight. So in this condition, sometimes some people क्या करते हैं, कान को पकड़ के उठा लेते हैं. This is not proper technique. We should holding its back side and pelvic regions, and it is the best technique to transfer from one cage by using both hand and hold the complete hindquarters. By using both hands, hold the complete hindquarters. These are the two techniques for holding of rabbit. And our main motto that during handling, the animal should not suffer with any type of pain, because if we will give any pain, then this will leads to development of release of some chemical that may interfere with our experimental data, or the animal will be irritate. Animal will be irritated if we will give pain to. अगर मैं आपको पेन दूंगा, आप इरिटेट हो जाएंगे. तो our technique should not produce any pain, and it should be firm. And this is the technique for handling of guinea pig. In case of guinea pig, they are very humble rodents. From beginning, I am telling that guinea pig are very humble, docile, and vegetarian. And they sometimes take chow diet. And if the animal is taking chow diet, then you should give vitamin C. And you should mix the vitamin C with their diet, so that the deficiency of vitamin C can be minimized. Otherwise, they may infected with some infectious disease and may die. And here, what we will do, we have to use both hand under the chest and use your other hand to support its hindquarters. Two figure representing. This is the technique how we can hold the guinea pig. It is our animal handling section. In another section, we will discuss about various routes of drug administration. It is also essential part, because most of we work on animals, and we should know how we will administer drugs and what should be the size of needle for administration of drug. Most generally, for extract, most of us work with some plant extract. In that cases, we give the drugs through oral route. Generally, most of we use mouse, rat. For oral administration of drug, we should use oral gauge needle. And for mouse, it's 24 to 20 gauge, and the length should be 2.5 to 3.8 cm. And ball diameter, it is essential. Sometimes we have observed, हम कुछ कभी कभी देखे हैं कि हम रेड का नीडल को हम क्या कर देते हैं, माउस में यूज कर देते हैं. This is the wrong practice. With the needle size for mouse is different, and needle size for rat is different. And for rat, we should use 20 to 16 gauge, and for mouse, it is 24 to 20 gauge. And length is also different, because there are some criteria how we will determine the length. We will discuss later कि हम length, needle का length का size क्या होना चाहिए. Length of the needle is 2.5, most in the range in between 2.5 to 3.8 centimeter, and for rat it is 3.8 to 10 centimeter. But 10 centimeter is too much lengthy, and we will see in next slide that how we will determine the length diameter. And ball diameter is 1.25 to 2.25 millimeter, and 2.25 to 4 millimeter. Volume always, it is important thing. Sometimes students asked कि सर मैं इतना डाइल्यूशन बनाया हूँ, कितना डाइल्यूशन बनाना है, कितना है. They prepare dilute, very dilute solution, and it will be difficult. That animal may die. This is the very essential things. We should always focus on this thing. Our solution should be such that it should be less than 10 ml per kg. It should be always less than 10 ml per kg, not gram. 10 ml per kg, 10 ml per 1000 gram, and in case of red, it should less than 10 ml per kg. And here, 20 gram, the maximum volume should be 0.25 ml. Maximum volume should be 0.25 ml. Otherwise, the animal may die. Animal may die. And for red, it should maximum volume 2.5 ml. Oral administration. It is very essential point. We should always note down it. For mouse, our quantity should not be more than 2.5 ml, 0.25 ml, and for red, not more than 2.5 ml. And these are some metal gauge needles, plastic gauge needles, and this is rubber feeding tube for reds. Sometimes some people use rubber feeding tube, so rubber feeding tubes, and sometimes we use metal gauge needle, plastic gauge needles. So here we will study one by one how drugs are administered. For intramuscular route, one thing, always maintenance of pH. Generally, we not determine pH sometimes. pH range, it is very much essential. It should always be in 4.5 to 8 for rodents or experimental animals. And disposable gloves, these are all things you can use. And for intramuscular intramuscular route, hypodermic needles, that is 26 to 30 gauge, maximum 0.3 ml. Some important points we should note down, that hypodermic needle we should use, that is 26 to 30 gauge, and maximum volume 0.3 ml. More not, so your drug volume should not more than 0.3 ml. And pH, always you adjust the pH. That is very much essential. It is strongly recommended that your pH should be adjusted, maximum volume should be fixed, and the needle size is fixed. And intramuscular, while administering drug from intramuscular side, rarely we use the intramuscular route for administration of drug, or if needed, when needed, we should use on thigh muscles of hind limb. Another important thing, that if there is chances of damage to sciatic nerve. This sciatic nerve is present with us also. It is also present in animals. And if we will make any mistakes for administration of drug and your nerve will damage, then that animal become paralyzed. So while administration of drug through intramuscular route, we should follow that such technique that sciatic nerve should not damage, which is located in the close proximity to femurs. It is located in close proximity to femur. This figure is representing the site for intramuscular administration. And restrain the animal by grasping the skin along its back with your left hand. If right-handed, administer substance in steady fluid motion. Take care not to administer fluid too rapidly. You should not administer too rapidly, and you should administer very slowly. And otherwise, it will make necrosis and may problem to that animals. Here, that things, the sciatic nerve damage, we should be very cautious that our needle should not damage sciatic nerve. Sciatic injuries is a typical task here. So these two things we have to remember while administering drug through intramuscular route. Another, subcutaneous injection. Very less use. Some intramuscular very less use. Subcutaneous injection, while giving the drug through subcutaneous, here we may observe how we administer a drug through subcutaneous route. And here we also need needle size 26 gauge and maximum 5 ml per kg. Always in my presentation, I am representing the maximum volume, needle size. It is essential that is size of the needle, hypodermic needle, 26 gauge, and with all of we know the subcutaneous, what is the subcutaneous side, and what should be the maximum volume, 5 ml per kg administered, and subcutaneous injection in the space between skin and underlying the muscles. Here we देखो हम यहाँ पे क्या किए, ये स्किन को उठा दिए हैं, तो here the space will appear in between, the inside the skin, and the one space will appear. In that space we will give the injection. Restrain the animal by grasping the skin along its back with your left hands. intercapsular or inguinal area, administration in the loose skin. In this, in the intercapsular, the inguinal area, the two sides are there in the rat or mice. Here we administer through subcutaneous route. Intradermal side, here in these slides we can observe the while we give the intramuscular 90° and subcutaneous for 45°, intravenous less than 25°, and intradermal less than 10 to 15°, in between 10 to 15°. So epidermis, dermis, subcutaneous, and muscles. So while we give the injection, intradermal, the angle of the needle should be less than 10 degrees Celsius. It is administered between the layer of the skin, and it is very typical work. And always we should perform all these experiments under the supervision of any expert person. The animal must be under general anesthesia for this procedure, and the 10 to less than 10 to 15° angle as on pictures. And here the needle size is 25 to 30 G, 25 to 30 gauge, and the total volume is 0.1 ml. Here total volume is 0.1 ml. In previous slide also we also observed 0.3 ml. Here for intradermal injection, the maximum volume 0.1 ml. It is very much essential, and every pharmacy professional, students, teachers should know these things while they work on animal. And if they it is very much essential, and they should know these things. Another, intraperitoneal. Generally we perform this, we inject the drug through this route, intraperitoneal route. Generally most of the time we use this route, and it is very easy. And here we use 22 to 30 gauge needle, 22 to 30 gauge needles, and maximum volume for rate is 1 to 3 ml, and maximum for 1 to here the maximum volume is 5 to 10 ml per kg. And here the site, यहाँ पे एक प्रॉब्लम्स क्या होता है, जब हम intraperitoneal route में administration करते हैं, there is chances of damage to some vital organ because the kidney, liver, and other organs are available here. So how we will confirm that our needle not damage to that vital organs? If while जब हम prick करते हैं और उसको हम remove करेंगे, तो if the blood will appears in this area, then there is chances of damaging of that vital organ. So while giving through intraperitoneal route, while administering drug through intraperitoneal route, we should be cautious that our needle should not damage any vital organ present inside the body of the rat. That is, there is peritoneal cavity is there, and peritoneal fluid is there. To that peritoneal cavity, drug to that peritoneal cavity, not the muscles, not to any organs, because this needle will go to and damage to any internal organ. The animal may die within some days. So we should be cautious that our needle should not damage any vital organ present inside the body of that experimental animals. That is lower left or right quadrant of abdomen, left quadrant or right quadrant of abdomen. That is the site for administration of intraperitoneal injection. Intravenous injection. It is sometimes we give our drug through intravenous route. What will generally we need restrainers. Sometimes we need restrainers, and the needle size is 25 to 30 gauge, maximum 0.5 ml. Here the maximum we can give 0.5, maximum 0.5 ml injection. Injection site lateral tail vein. Injection site is lateral tail vein. Important thing, the name of that vein is lateral tail vein, and we should make prominent by giving some venodilators and lateral tail vein, and restrain the animals by mechanical restrainers. And without restraint we cannot perform this work. With the tail on slight tension, and insert the needle at least 3 mm into the lumen of the vein. At least 3 mm into the lumen. Here one thing we always remember, that you should not aspirate the syringe. You should not aspirate the syringe, otherwise the vessel will collapse. Generally we aspirate sometimes. We should not aspirate here. Otherwise, the vessels will collapse. The vein will collapse. Slowly, fluid motion to avoid rupture of the vessel. In all this work, all while administering drugs, always we should perform very slowly, not be fast. समटाइम्स क्या हो जाता है, हम हड़बड़ी में रहते हैं, जल्दी जल्दी इंजेक्शन दे के निकल जाए, वो नहीं होना चाहिए. वी शुड बी very slow, very slow while injecting the drugs, and we should not aspirate while giving the intravenous to the experimental animal, and remove the needle, apply pressure, stop bleeding, returning animal to cage. तो क्या, अगर हम ये एक्टिविटीज को परफॉर्म रेगुलरली, जो भी फार्माकोलॉजी पढ़ाते हैं या फार्माकोलॉजी स्टूडेंट्स हैं, तो उन्होंने उसको रेगुलरली परफॉर्म करेंगे या एक उनका शौक में करेंगे, और प्यार से एनिमल हैंडलिंग जब करना होता है, तो हमको एनिमल के साथ वी शुड अफेक्शनेट विद एनिमल, वी शुड बिहेव ह्यूमनली विद एनिमल. हमको एनिमल के साथ ह्यूमन बिहेवियर करना है. Otherwise, you cannot perform any experimentation with animals. हमको एनिमल को पशु जैसे नहीं समझना है, हमको एनिमल को एक मानवता की, जैसे मानव जिस इंसान जैसे समझना है, हमको उस एक्सपेरिमेंट परफॉर्म करना है, so that we can easily perform our experiment. पेरेंटली नहीं यूज कर सकते हैं, तो उस कंडीशन में हम क्या करते हैं, यूज करते हैं. और ओरली, जब ओरली व्हेन वी एडमिनिस्टर ओरली, सो फिगर रिप्रेजेंटिंग द दिस लिंक ऑफ द ओरल फीडिंग नीडल. ओरल फीडिंग नीडल जो है, ओरल गेवेज नीडल जो है, ये लेंथ कितने होना चाहिए, व्हाट शुड बी द लेंथ ऑफ द ओरल गेवेज नीडल रिप्रेजेंटेड बाय दिस फिगर. It is essential. It is very much essential because different sizes of oral gauge needle are available in our laboratories. लेबोरेटरी में बहुत सारे, बहुत प्रकार का होता है, तो हमारे कंफ्यूजन ये होता है कि हम कौन से लेंगे, किस फीडिंग को मिल, ये बड़ा होता है क्या प्रॉब्लम्स है, छोटा होगा तो क्या प्रॉब्लम्स है. इफ विल साइज विल बी लार्ज एंड वी फोर्सली अप्लाई दैट needle, forcefully apply, it will damage some delicate soft part inside the mouth or inside the internal body of that small animals. अगर ये एनिमल्स के जो माउथ के अंदर में छोटे पार्ट्स हैं, उसको डैमेज कर देगा, दैट एनिमल में डाई. सो मेजर द डिस्टेंस फ्रॉम द टिप ऑफ द नोज टू द टिप ऑफ द नोज, द टिप ऑफ द नोज टू द फर्स्ट रिब. फर्स्ट you make the first rib. This is of needle that should be used. This is the length of the needle that should be used. Restrain the mouse by grasping the skin along that things we have discussed previously. How we will hold, and in that manner we hold that animal, and we measure the length of that needle, and then we collect our drugs and 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 give to that experimental animals. Here, one thing we should be very cautious that our needle is made up of steel. It should not damage hard palate or any mouth parts of that animal, and maximum dose should be 10 ml per kg. 10 ml per kg for oral administration of drugs. And I also in previous slides mentioned that कि नॉइस के लिए वॉल्यूम कितना होना चाहिए और रेट के लिए कितना होना चाहिए, प्रीवियसली उस स्लाइड्स में मैंने एक्सप्लेन किया है. दैट थिंग्स you should always note down. And next is very essential part, anesthesia techniques in rats, क्योंकि it is essential part, and most of the times we perform anesthesia on experimental animal. देखिए हमारे जो लेबोरेटरीज फैसिलिटीज मोस्ट कॉमनली क्या है, कुछ से बहुत सारे जगह में इसका स्पेशल फैसिलिटीज अवेलेबल नहीं होता है. इंडक्शन चेम्बर, ऑल दीज आर नॉट अवेलेबल. सो इन दैट कंडीशन वी टेक सम बेल jar or some glass chamber फॉर इंडक्शन ऑफ एनेस्थीसिया, उस कंडीशन में हम क्या करते हैं, कुछ बेल जार्स ले सकते हैं, एंड for induction of anesthesia, and we should be very cautious, otherwise animal may die. And generally use chlorellose, urethane, barbiturates, paraldehyde, magnesium sulfate, ketamine, tribromoethanol, isoflurane. Numbers of anesthetics are available. So as per our need we select, and some anesthetics may possess some toxicity, so keeping in mind that things, we should select our which anesthetics will be better for our study, and then we perform the experiments. And anesthetics sometimes, जैसे कि most people ether use ether, and this may causes death of animals. And chloral hydrate कॉमन इन न्यूरोफार्माकोलॉजी। आए बारबिचुरेट्स हम लेते हैं, बारबिचुरेट्स में क्या करते हैं, बारबिचुरेट्स ऑल ऑफ वी नो दैट बारबिचुरेट्स आर हैविंग नैरो मार्जिन ऑफ सेफ्टी, इट मे कॉजेस डेथ ऑफ दैट एनिमल, सो ऑलवेज we should fix the dose, finalize the dose, then we go for that induction. And isoflurans, reasonably research anesthesia and more expensive than halothane and require own vaporization, no concentration at no mortal concentration at room temperature. Isoflurane can use. And for induction anesthesia, some is here the chart is available. For duration of ketamine induction by injectable anesthetics, sometimes we use injectable anesthetic, sometimes we use wafers. So for injectable ketamine, 15 to 20 minutes for ketamine, 75 to 100 mg per kg, IP route. You can give through IP route, and ketamine plus xylazine, 6 mg per kg, 5 to 10 mg per kg, and ketamine plus thomidine, 30 to 40 minutes, 25 mg per kg, plus as per our need, as per our duration, how much we need to anesthetize. Uh, we have to select the anesthesia, and we have to select the techniques. And another most important part is blood collection techniques. एनेस्थीसिया हमको कब जरूरत पड़ता है, जैसे हम कोई परफॉर्म कर रहे हैं, जैसे कि ड्रग एडमिनिस्ट्रेशन कर रहे हैं, या अभी कभी कभी हमको क्या है, कुछ सैंपल कलेक्शन करना है, एनिमल इज वेरी मच एग्रेसिव, और इसी कंडीशन में, इन दैट कंडीशन वी यूज anesthetics. And which sometimes anesthetics may interfere with our result. We should keep in mind which anesthetics we should use, otherwise it interfere with our experimental data. Blood sampling in mice are red. It is most important parts कि how we'll collect the blood samples. हम जितने भी एक्सपेरिमेंट करते हैं उसमें blood sample, it and various parameters, it's blood sample, and most of us generally collect the blood sample. Generally we prefer tail vein, orbital sinus, cardiac punctures, saphenous veins. So all these are most important site for blood collection. So first thing, while getting the approval for experimentation of on animals, we take the approval from CPCAC. किसी भी एक्सपेरिमेंटेशन करने से पहले सबसे पहले हमको हमारा रेस्पॉन्सिबिलिटी होता है कि we should take the approval from CPCAC. Without getting any approval, we should not start our experiment. ये फार्माकोलॉजी ना भी कोई एक्सपेरिमेंट सकते हैं, आप for the CPCAC approval, IAC approve our protocol, institutional and committee approve our protocol. At that time, you must describe your protocol, how you will collect blood from that animal. कलेक्ट करेंगे, आपको ये इंफॉर्मेशन होना चाहिए, हमको पता होना चाहिए हम कैसे ब्लड कलेक्शन करेंगे, कहाँ से ब्लड कलेक्शन करेंगे. From beginning, who are performing any experiment, our should not produce any type of pain to anyone. हमारे सारे एनिमल पे इंटरवेंशन का law है कि हमारे जो भी should always for the benefit of the society. It should be less painful. Adequate training is required. एक पॉइंट मैंने पहले से बोला था कि अगर if we don't know the technique, we should perform our work under the supervision of any expertise. मतलब हमारे डिपार्टमेंट में हो या नियर बाय डिपार्टमेंट में हो, जिसको जो पर्सन एक्सपर्टाइज है हम उनसे कंसल्ट करें, देन वी स्टार्ट आवर एक्सपेरिमेंट, उनसे गाइडेंस लें, अदरवाइज द एनिमल में डाई। क्या होता है, वी मेक एरर इन डिफरेंट टाइम्स, समटाइम्स वी नॉट गिव द माने कंसंट्रेट ऑन वॉल्यूम ऑफ द ड्रग्स, कितने वॉल्यूम हो रहा है हम उसको ध्यान नहीं देते हैं। समटाइम्स वी मेक एरर ऑन डोज कैलकुलेशन, वेरी कॉमन मिस्टेक वी डू, the dose calculation, volume. We always keep in mind all these things. Otherwise, we will not get the proper result, and our work will become worthless. And, frequency is very important. While collection of blood, frequency is also important. We we don't know कि हम कितने बार कलेक्ट करना है, कितने बार कलेक्ट कर सकते हैं। अगर ज्यादा कलेक्ट करेंगे तो क्या होगा, हमको बहुत सारे लोगों को पता नहीं होता है। Once in 2 week is ideal for rodents. Once in 2 week, once in 2 week, in 14 days में एक ही बार rodents के लिए है। If we want to collect a large quantities of blood, if देखो हम बहुत बहुत blood glucose level measurement करना है by using kit, that is different matters. If you want to collect, हम ज्यादा वॉल्यूम क्वांटिटी में ब्लड कलेक्ट कर रहे हैं तो once in 2 week इज आइडियल। ये डिपेंड करता है आपके वर्क में आप क्या कर रहे हैं। और इफ यू नीड टू मल्टीपल ब्लड सैंपलिंग, एट दैट टाइम यू कैन यूज लार्ज एनिमल, यू कैन गो फॉर रेबिट, यू शुड नॉट सिलेक्ट रोडेंट्स, रोडेंट्स आर वेरी स्मॉल। हम रेट या माइस में ज्यादा अमाउंट ऑफ ब्लड हम विद इन 1 week 2 times कलेक्ट कर दिए तो इट विल इफेक्ट टू animal health. So in that condition we should select rabbit, and because the blood quantity is more in comparison with rat, so हम rabbit को replace कर सकते हैं, because some diseases rabbit can also give the good result. So this figure and this slide representing how we will collect blood from tail vein. फ्रॉम टेल वेन से हम कैसे ब्लड कलेक्ट कर सकते हैं, अप टू रिकमेंडेड फॉर कलेक्टिंग अप टू 2 ml, नॉट 2 ml, up to 2 ml means up to 2 ml, that is maximum quantity is 2 ml per withdrawal, maximum quantity, not more than that 2 ml, and 23 gene is inserted into the blood vessel, collecting using syringe. Complete blood collection. After collecting the blood, we should give pressure and we should apply some ointments, some antimicrobial ointment we can apply, or if you need to multiple blood collection, you can use the cannula. You can use the temporary surgical cannula you can use, but it is generally the people जो भी रिसर्च में रेगुलरली इन्वॉल्व रहते हैं या उनको फ्रीक्वेंट ब्लड लेना है वही करते हैं, लेकिन बहुत कम टाइम हमारे जो एकेडमिक में जो रिसर्च होता है स्टूडेंट्स जो करते हैं वहाँ पे हम कैनुला यूज करते हैं। हम मैक्सिमम उसमें क्या कर सकते हैं 2 ml फॉर विथड्रॉल, मैक्सिमम 2 ml फॉर विथड्रॉल, एंड ब्लड कलेक्शन हमारा टेल स्निप। दिस मेथड मैंने पहले वो तो यूज हो रहा था, लेकिन द यूज ऑफ दिस मेथड इज वेरी लेस, ओनली इन माइस। दिस मेथड ओनली रिकमेंडेड फॉर माइस, नॉट इन रेट। द टेल स्निप मेथड, इसमें क्या करते हैं वो कट द टिप ऑफ द टेल, this is called tail snip method, only for tail, and for when required very minute quantity of blood, when we require very minute quantity of blood, in that condition we can use tail snip method. And it's a painful method also, even because we cut the tail, and here there is chances of permanent damage of the tail, permanent damage, ये tail का permanent damage भी हो सकता है, and we should perform under anesthesia, local anesthesia we apply, and cut 1 mm from the tip of the tail. Blood flow is stopped by daving the tail tip. We should apply some drug molecule, some chemicals, and stop the bleeding. Otherwise, large quantity of blood may flow from the animals. Very less frequently it is performed, and tail snipping methods, and it is performed in mice because their tail are very thin and delicate. आप हमको एक चीज ध्यान रखना है कि हमारा किसी भी एक्सपेरिमेंटेशन में एनिमल का अननेसेसरी टॉर्चर नहीं होना चाहिए, एनिमल को ह्यूमनली बिहेवियर होना चाहिए। ये तो एक, हम एज ए ह्यूमन बीइंग, as a pharmacy students, as a pharmacy scholars, we should know all these things. And another method is the retroorbital science, very commonly used method. We collect the blood when we required blood. Generally we use retroorbital sinus. Under इसमें in this thing, in these methods, we use general anesthesia or topical ophthalmic anesthesia. Topical ophthalmic anesthetics. Topical ophthalmic anesthetics you can apply, or you may perform some general anesthesia. Otherwise, animal may not allow you, or animal become irritating. Another thing where we use anesthesia, because pain can be reduced. Pain can be reduced, and the animal is scrubbed with thumb and forefinger. Here we in figure you can observe thumb and forefinger. In previous slide I discussed how you will scrub how you will hold the animal thumb and forefinger and a capillary medial canthus of eye. This side is called medial canthus, and in this side and rotate it. The angle is 30 degree, not on the other side. Next slide I will show medial canthus of the eye, and the angle should be 30-degree angle, and one plexus punctured blood will come. Here the veins is there, and the plexus will puncture. Here the plexus will puncture, blood will come, and 30 minutes check the periorbital lesions. We should here for periorbital lesions. Sometimes what happens, your movements of capillary may damage to the other parts. It may causes lesions to that periorbital space. So here you have to check whether they are in lesion to this area. Here another thing that this figure is representing in the best way. देखिए, अगर if हम इस तरह से हम उसको puncture करेंगे, it may damage to that orbital parts or damage to that eye structure of that animals. So here this is the best figure, this is the best side. In this side we will puncture the orbital sinus, and here we retroorbital plexus. From this side we can collect the bloods. And if we will not perform in proper way, these things may appear, these things may occur. So हम अपने experiments के लिए for our benefit, how we are cruel and how animals are helping with us, to keeping all these things in mind, we should perform our experiments, our work on animal, and we should also behave on that animal. देखिए, हमारे career के लिए, हमारे development के लिए, हमारा सोसाइटी के डेवलपमेंट के लिए हम कितने एनिमल को यूज कर रहे हैं, तो उसी चीज को माइंड में रखते हुए हमको प्रॉपर वे में कि हमारे एक्सपेरिमेंट, ड्यूरिंग आवर एक्सपेरिमेंट्स द एनिमल शुड नॉट बी harmed. Our experimentation should not produce any unnecessary, unethical pain to that animals. So if these things may appear, if we properly not perform this work, so these things may appear. It will damage to that eye part of that animals. Blood collection by cardiac punctures. Here we use a needle size 23 to 25 G in mice and 19 to 21 gauge in rat, and makes 10 to 20 ml syringe. This figure is representing how you can collect, how this is the animated figure, and this is a real picture. And from this figure we can know that how you can connect, how you can collect the blood taken from the heart. For terminal blood collection, generally this blood collection from cardiac puncture is carried out for terminal blood collection. Terminal stage मतलब आपके एनिमल का और कुछ यूज नहीं है, sacrifice करना है. For that cases we can use this cardiac puncture techniques. Should perform under anesthesia. We should, and if the animals having dextrocardia, sampling may fall. In that condition, we should not perform. Blood collection saphenous veins. This is the lateral saphenous veins. From these saphenous veins, we collect blood, and that is the needle size should be 23 gauge needle, and collected by using capillaries. Collecting more than 4 sample in a day, 24 period is not advisable. Another most important part is euthanasia. Sorry, blood sample collection in rabbits. In rabbit, we use marginal ear veins or from artery. Marginal ear veins or artery for collection of bloods, and it is in case of rabbit, this side is prominent. This side is prominent. From this side, we can collect the bloods. So marginal anesthesia is not required. When easily accessible, topical anesthesia recommended, yield small to moderate. And total, what should be total blood volumes? In case of rabbit, 6% of lean body weight, 6%, 1% of body weight for every 2 weeks. And 2 kg rabbit 20 ml maximum, 4 kg of rabbit 40 ml maximum. In case of guinea pig, we collect the blood from saphenous veins and lateral saphenous veins, and for terminal cases we can use cardiac. The saphenous vein we have seen here, saphenous vein at a lateral saphenous vein. For the, it is easy to collect blood from the saphenous vein, but it is a typical task, and we should perform this work under the supervision of an expert. Euthanasia means the killing of animal used for scientific purpose. फ्रॉम बिगिनिंग, शुरू से मैं बोल रहा हूँ कि एनिमल को हम साइंटिफिक पर्पस के लिए यूज करते हैं, और यूथैनेसिया जो एंड आफ्टर द एक्सपेरिमेंटेशन, व्हेन द एक्सपेरिमेंटेशन पार्ट इज ऑलमोस्ट ओवर एंड वी नीड सम इंटरनल बॉडी पार्ट्स, हमको लीवर चाहिए, किडनी चाहिए, ब्रेन चाहिए, इन दैट कंडीशन वी परफॉर्म द यूथैनेसिया ऑफ दैट ऑन दैट एनिमल्स. Euthanized in laboratory or breeding establishment for following reason. What is the reason? Terminal studies when pain, distress, suffering. जैसे कि animal को हमको लग रहा है कि वो as a human being we know that how the animal is suffering, how the pain to that animal. In that cases we perform euthanasia when the health or welfare animal are ground for concerns. When animals are no longer used for breeding, when stocks are not required for certain reason, in that condition we perform euthanasia on animal. So some techniques here available. Rates: physical method, inhalation of gases, and drug administration. Physical method में क्या कर सकते हैं? Exsanguination, decapitation, cervical dislocation. Generally it is performed cervical dislocation, decapitation, exsanguination. And here, if the weight of the rat is more than 150 gram, we should use anesthetize. We should anesthetize first, and then we perform the euthanasia. Inhalation of gases, carbon dioxide plus oxygen mixture, 70%, 30%, and optimal flow 20%. Chamber volume produced by isoflurans, and drug administration, barbiturate overdose, IP. If you will give barbiturate overdose, euthanize automatically, it will animal may die. And for guinea pigs, chemicals may use inhalants, carbon dioxide, halothane per percent, injectable pentobarbital sodium, 90 mg per kg, and physical cervical dislocation. And for rabbit, inhale halothanes, injectable pentobarbital sodium, IV, IP, 60 mg per kg, and captive blood neck dislocation or decapitation following anesthesia. Thank you for your patience and listening. And if you are having any questions, please.
Participants may ask their questions. Thank you, sir, for sharing your deep knowledge with us. It will be a great pleasure for us to listen to you again in our future events. Thank you once again, sir.
Thank you.
Tomorrow we'll start our session sharp at 10 a.m. I once again thank you all for your active participation. Thank you.